Well, hello everybody. Today I'm going to talk about genetic affiliations of populations of Agassiz desert tortoises who live in the western Sonoran Desert, specifically the Coachella Valley of Southern California. This area is in the southwestern extreme of the range for the species and is probably better known today for its namesake music festival, but some of you may also be familiar with towns like Palm Springs, Palm Desert, Indio. These are towns in the Coachella Valley. I am not a geneticist, but I collaborated with some very talented geneticists on this publication, and the results are already published in early view in the next issue of the journal Frontiers of Biogeography. So if you're interested in details beyond what I give in this presentation, you can check that out. I've been studying a population of desert tortoises in one of the many wind farms near Palm Springs, California for over 25 years now. And the population is isolated from other tortoise populations. And I've often wondered what their relationships are and what their affiliation is genetically. And that was the impetus for this study. Now, tortoise genetic connectivity is known to be affected by both ancient and modern topographic features, and I'll give examples of both. But high mountains and low, hot valleys within their range are definitely barriers to dispersal. There are also aquatic barriers, as have been shown in previous publications, things like the Colorado River and one of its aquatic precursors that happened in ancient history that I'll talk about. But even human features at the localized scale, like roads and railways, have been shown to affect genetic structure in tortoises. Now, this is a map that shows the range of Agassiz desert tortoise in the southern part of its range, and locality dots are shown scattered throughout. But notice that there's a division in the distribution down here in the southwest portion of the range near the Salton Sea. What causes that split? Well, there's a low-lying area called the Salton Trough that's like a wedge that moves northwestward from the shore of the Gulf of California in Mexico. And this is a very low elevation area that's unsuitable as habitat for the tortoise. But tortoises did manage to get to the other side at some point in time. And one of the study sites I'm going to talk about and call my Western study site is shown here at the tip of the Salton Trough. These populations are also isolated somewhat from major recovery actions that are trying to help the species recover from its status as a threatened species under the Endangered Species Act. And you can see the nearby critical habitat for the species shown in yellow. And so these tortoises are missing out on some of the recovery actions that occur in those areas, despite the fact that they're still protected under the Endangered Species Act. So this is a view of the Coachella Valley. It's low, it's hot, it's sandy in many areas, much of it's below sea level, and it's in the western Sonoran Desert portion of California. It's arguably the hottest and most hyper-arid portion of the Sonoran Desert. As a result of that, it has a very short growing season, extremely high summer temperatures, and is overall unsuitable for tortoise habitation. Now, the geography of this area, the desert southwest in particular, is best described as tortured. It sits at the interface of the North American and the Pacific plates, so there's lots of tectonic activity and shifting and shearing and twisting and turning. It's also been flooded by marine incursions over time that I'll show you examples of. The uplift of the transverse ranges extend just across the northern boundary of this area, and they are over 11,000 feet high, but they were uplifted within the last 2 to 5 million years. The uplift of the Colorado Plateau, which is to the northeast of this area, occurred over about the last 6 million years. It's over 1 mile high. And then the evolution of the modern Colorado River. It didn't always flow to the Gulf of California as it does today. Now, I'm not going to belabor these, but I want to point out a couple of things. One is that 6.3 million years ago, the whole area of the desert southwest, including the Salton Trough, was flooded by the Gulf of California. PS shows the location of Palm Springs today, and you can see that there was a giant estuary that extended far into what's now the Mojave Desert. And this would have been a significant barrier for movement of plants or animals that may have been limited by this feature from east to west or even north to south. And so this had an important effect on the evolution of the species that we see there today. And then only 5.3 million years ago, the Colorado River actually flowed south through this area and joined the Gulf of California into its present course today. But that was not the case prior to that event. And of course, the presence of this river also provides a barrier or a filter for plants and animals that live in the area today. Biologists are very interested in all the geological changes that occurred in this area over the last 6 million years. And they've studied many different species of lizards and snakes and plants and tortoises, trying to figure out the timing of some of these events as precisely as possible. Here's another example showing the Pliocene extent of the Gulf of California, also known as the Sea of Cortez at that time. And you can see again that it penetrated deeply into what's now the Sonoran and Mojave Desert portions of Southern California and Arizona. And this would have had dramatic effects on isolating species on either side of these major aquatic barriers if they couldn't fly, swim, or jump over them. In fact, they're implicated in the timing of the split between the Agassiz desert tortoise and Marofka's desert tortoise, now found basically on opposite sides of the Colorado River. And the estimated timing of this geological formation, known as the Baus Formation, is concordant with molecular clock estimates of mitochondrial DNA evolution that led to the separation of these species. So we sampled 3 tortoise populations on 2 sides of the northern part of the Salton Trough, which is called the Coachella Valley. There's the Coachella Valley in light gray. We had our western study site and then 2 eastern study sites, and the distance between them was about 85 kilometers. For tortoises to move back and forth between these sites, they would have either had to cross the northern Coachella Valley or go through this very rugged terrain of the Little San Bernardino Mountains, which are part of the transverse ranges, and they're over a mile high. Give you a quick snapshot of each of the study sites. The western study site, like I said, is in an active and operating wind energy facility. It's an interesting ecosystem because it has elements of Mojave and Sonoran desert plants, as well as some montane and coastal species from Southern California. So it's quite different from other areas in the desert tortoise's habitat. Eastern study site number 1 is located within the protected southern boundaries of Joshua Tree National Park. This area has typical, for California, Sonoran desert vegetation with visually dominant sub-tree species like ironwoods, palo verdes, smoke trees, and then ocotillos. So very, very characteristic of California's Sonoran desert. Eastern study site 2 is nearby, and it's more of a creosote bush flat with some of the scattered tree-like forms that I mentioned at the nearby site previously. So we took blood samples from tortoises that resulted in 53 DNA samples from those 3 study sites. We analyzed mitochondrial DNA for Eastern Study Site 2 and the Western Study Site just to establish a baseline for populations and see if we had any evidence of translocated tortoises dropped off from faraway places, because that has been documented. And then we genotyped all the samples using the microsatellites shown following the procedures previously published. We had a reference database of over 700 samples of tortoises from throughout the range of Gopherus agassizii from these prior studies, and we used those to compare our 53 samples to using assignment tests and structure analysis using the genetic units that were identified by Sanchez Ramirez et al. That I'll show you. So here they are. The ones that were of interest to us because of their proximity to our study sites were Western Mojave, Central Mojave, Southern Mojave, and Colorado Desert. Significantly, nobody has ever previously analyzed tortoise samples from the southwestern extent of their distribution, so that's where ours come in. We also sampled some tortoises from the Santa Rosa Mountains in the southwestern part of the Coachella Valley. Those results won't be included here. We hope to use those in the future for comparisons with tortoise population a little further south in Anza-Borrego Desert State Park. So for our mitochondrial DNA results between Eastern Study Site number 2 and the Western Study Site, all the individuals had haplotype Mojave A that's found throughout most of California. There were no surprises, no ringers. We didn't find any Gopherus marofki. We didn't find tortoises from the northeastern Mojave or the far western Mojave. it appeared that the tortoises we were working with all had the expected haplotype for tortoises native to that part of the range. Then looking at assignment tests, our Western study site was split in assignments between the Southern Mojave and the Colorado Desert, although the majority of them aligned with the Colorado Desert recovery unit, which is shown in yellow in the small figure below. But then the Eastern study sites 1 and 2, which we combined in analyses overall, were aligned with genetic samples from the Colorado Desert, which is not unexpected because they're the closest to that genetic unit. But these results will have significant impact as I get to the conclusion. Then we did a structure analysis, and remember we had 709 reference samples plus R53, and each line in this histogram shows one of those samples, and then using statistics to identify which population structure was best fit our data. We did it for, we asked, what if we assume we had 2 populations? How would the data look? What if we had 3? We did all the way up to 12. And statistically, a K equal 5 or 5 populations had the best fit to our data. And so you can see it, there's a clearly identified Western Mojave genetic unit, a clearly identified Central Mojave unit, et cetera. It gets a little mixed up in the Southern Mojave with Lucerne Valley, but that wasn't part of our study area, so we're not really concerned about that. But all 3 of our samples aligned with the Colorado Desert genetic unit, sites 16, 17, and 18. And it's important to note that the Western study site had 75% affinity with the Colorado Desert genetic unit, despite its geographic distance from there. And it actually fell out at K equals 7. If you assume that there were 7 populations and you ask it to redraw the histogram, it would have dropped out as separate. Just a few alleles, very minor differences with the others. So basically, this is a remap of the Sanchez Ramirez et al. Genetic units that were identified, and they initially demarcated the Colorado genetic unit as occurring east of this dashed blue line that I'm trying to highlight with my mouse right now. Our study site 16 is Western study site, 17 and 18 are the Eastern study sites. And we hypothesized that the relatedness between these is due to the possibility that tortoises are still able to maintain connectivity along the foothills of the Little San Bernardino Mountains, avoiding the hyper-arid, low-elevation northern Coachella Valley. So that's likely explanation for that relatedness. And then there was a relationship, like I showed in the assignment test, between the Western study site and the Southern Mojave, and there are canyons in this area called the Morongo Canyons that connect these 2 ecosystems, joining the Sonoran to the Mojave Desert, and that's the most likely reason for that. So this is kind of a mixture of the 2 groups that it sits next to. And so we actually recommend in our research that the genetic unit for the Colorado Desert should be extended all the way up here to what's called San Gorgonio Pass to encompass the genetics of these tortoises. A paper was recently published showing the importance of mountain passes for genetic exchange in tortoises. Our data suggests that this is occurring there between the Western study site and the Southern Mojave Desert. So we conclude that the Coachella Valley does not appear to be a significant barrier to genetic connectivity with the Colorado Desert genetic unit of tortoises, that mountain passes are allowing that connectivity between Mojave and Sonoran tortoises in San Gorgonio Pass near Palm Springs. And then finally, we conclude that the Colorado Desert genetic unit should be extended to San Gorgonio Pass to recognize that relatedness. A number of groups help with this research and funding and on-the-ground sampling, and they're listed here. And if there's any time for questions, I'd be glad to take them.